In order to study viruses, we've got to be able to grow or cultivate them. Since viruses can't copy their own DNA or RNA, they must have a living cell as their medium. So that gives us the need to have special methods like inoculation of animal cells or bird embryos. All right. So this is kind of an interesting thing we have to do. We can't just grow them out on a plate of agar necessarily. All right, so what techniques do we use? Well, then they require living cells as their medium of growth. And so we can categorize this in two ways, in vivo or in vitro. So in vivo is going to be a laboratory bred animal or embryonic bird tissue, an embryonated cell. In vitro is going to be in an actual cell or tissue culture method. So instead of an embryo, it is just a cell or a piece of tissue. And so the reason that we want to cultivate a virus is so that we can isolate and identify viruses in a clinical setting. We want to be able to prepare viruses for vaccines. This is very important. And this is something that is very relevant recently as we've had to make new vaccines for the coronavirus. This is the technique that had to be done in order to grow and prepare and make a vaccination, sorry, for the vaccine. We had to use an embryo to grow it in. All right. And so in order to do that detailed research on the viral structure, the genetics, the cycle that they multiply by, how they affect us, we've got to be able to grow them in order to study them. So sometimes we do this into a live animal, specifically bred strains of mice, rats, guinea pigs, rabbits. Those are usually what we use for viral cultivation. Sometimes we have to use invertebrates like insects or non-human primates. Viruses do have that host specificity, so that's what is going to determine what we use to grow or propagate the virus. And so they're going to do better in some species as opposed to others. All right, bird embryos. So chicken, duck, turkey eggs, these are the most common choices of bird embryos to inoculate. Viruses are going to be injected through the eggshell by drilling a small hole or making a small window. These are really handy because they're intact and self-supporting. It's a pretty sterile environment, and they also contain their own source of nourishment. So this is a really handy method. So using a tissue cell culture like a HeLa cell, this is going to be a growth in a sterile chamber that's going to have a special medium that has the correct nutrients needed for the cells to survive. Cultured cells form a mono or single layer of cells that support viral multiplication. And this allows for close inspection of culture for signs of infection. So here's our next NCLEX prep practice question here. Which of the following is a known association between viruses and cancers? How well were you listening to the last PowerPoint? All right, pause. All right, so is it A, papillomaviruses cause brain cancer? B, infection with herpes virus leads to AIDS? Hepatitis B is associated with liver cancer, or papillomavirus is associated with gastric or stomach cancer? If you've been listening and made your index cards with these guys and been studying those, you should know that hepatitis B is associated with liver cancer. HEPA basically in Latin means liver. So anytime you see HEPA, it is going to be somehow related to the liver. So how do we know the virus is growing? All right, and so this example is for things like if we were going to grow a bacteriophage. A bacteriophage can actually be grown on a lawn of bacteria in a petri dish. So one way to see viral growth in a culture is to look for the degeneration and lysis of infected cells in that monolayer. So these areas that have been destroyed show up as a clear patch, a defined area as what we call a patch. And so these plaques, did I say patch or plaque? These patches are known as plaques, a plaque, P-L-A-Q-U-E, not sure which I said. These plaques are what we call a visible cytopathic effect. All right, so we talked about cytopathic effects. So this, let me put on my little light here. This is what we would originally see. So this looks like a monolayer of cells. If I had to guess, this could possibly, maybe this is epithelial cells, okay? I said bacteria, but this is not bacteria. All right, so epithelial cells. And so this is what it looked like. We inoculated this with some type of virus. And so these clear areas are what we would call plaques. And we do see these on Petri dishes with bacteriophages and bacteria as well. That may have actually been in the chapter 2 lecture. It seems like that's been done somewhere already. All right, so here's what I was talking about initially. How would we detect bacteriophages? The same technique here is going to be used to detect and count bacteriophages because they also produce plaques when grown on agar as a lawn of bacteria. The infection will spread symmetrically around in the point of infection and give that clear space where the dead cells are, as we just saw in the other picture. Prions are really cool. These are infectious proteins. They're non-cellular infectious agents. These guys are smaller than a virus, and it's really just a protein that has become infectious, okay? No nucleic acid or anything. So these are some of the worst diseases that are actually the hardest to treat and get rid of. They're progressive. They're usually fatal. They all deposit protein fibrils in the brain. The exact mode of infection is actually still being investigated. And this occurs to humans and animals, all right? And so we have some examples here, some that affect humans, some that affect animals. Creutzfeldt-Jakob disease was the first that was discovered, and it affects the central nervous system and causes degeneration and death. Animals can also be infected by these, and you've probably actually heard of this, and you may not have heard it as this name, but bovine spongiform encephalopathy, and that is actually mad cow disease. And so this was a huge crisis in Europe, and if you are a little more seasoned like I am, you might remember this from the 1980s and 90s when it was found to be spreading to humans, and so they had to slaughter thousands of livestock. So this was a huge hit to the agricultural industry. It's abbreviated BSE. This was the first time that a prion was spread from an animal to a human, so that's pretty significant. In 2016, we have Shy-Drager syndrome, which is also called multiple system atrophy, or MASA, M-A-S-A. It was recognized then. It is very similar to Parkinson's disease. Proteins accumulate in the brain and cause symptoms throughout the body. Both Parkinson's and Alzheimer's have a similar accumulation of these protein fibrils or plaques in the brain. We call them amyloid plaques, sometimes beta plaques. And so this has led to the suspicion that there could possibly be a link to prion infection and those diseases. And so that is an area of research, a very interesting area. All right, so which of the following diseases is not caused by a non-cellular infectious agent? A, acquired immunodeficiency syndrome, or AIDS. B, tuberculosis. C, smallpox. D, Creutzfeldt-Jakob. Or E, measles. Pause, pick your answer. All right, so it is B, and you may be thinking non-cellular infectious agent was just the prions, but no, that's anything, remember, that includes viruses, right? So viruses or prions would fall into that category. So satellite viruses are virus-like particles that depend on other viruses for replication. Satellite RNAs do not encode the capsid proteins, but are packaged by a protein encoded in a helper virus genome. So the adeno-associated virus is an example here. It was named this way because it was originally thought that it could only replicate in cells with other adenoviruses, but it was later figured out that it can infect other viruses. The delta agent is actually a naked circle of RNA that is expressed with the help of hepatitis B. And so when these are what we call comorbid or occur together, this will worsen the liver damage caused by hepatitis B. Fibroids are basically very small viruses. They're 1/10 the size of an average virus. They do not have a capsid or any type of coating. These guys are a huge problem in agriculture because they can infect a lot of important crop plants. So they parasitize plants is what they do, all right? Particularly things like tomatoes, potatoes, cucumbers, chrysanthemums, citrus trees, all right? Some of those are among my favorites, right? Cucumbers, we don't want to do without those. There are many, many viruses that affect human health: hepatitis, chickenpox, influenza, colds. Common cold is caused by rhinovirus and coronaviruses. Of course, a lot of viruses are very prominent worldwide that we don't necessarily see as much here: dengue fever, Rift Valley fever, yellow fever. We have many that have really high mortality rates. Rabies is one of the absolute highest, and we'll get into that later on. AIDS, Ebola. We have many that can cause long-term disabilities, and thankfully, these we actually have immunizations for, like poliomyelitis and rubella. And then we have some that are really connected to chronic infections like multiple sclerosis and Alzheimer's disease, all right, and of course various types of cancer. I was hoping to find a nice little graphic that kind of showed all of these, but there wasn't really any. So I got one here on COVID. This is actually from May of 2021. It is impossible really to accurately measure worldwide viral infections because of lack of reporting. But anyway, so this is some of those here, or actually this is coronavirus here, new cases. Oh, I thought I had my little thing there. There we go. All right, so new cases here. This is February of 2021. And then it's gone down here. We know that I believe it went back up for a while. And so they do go up and down. All right, and then so here's some hard numbers on that data as well. Table 5.5 in the textbook lists the most common viruses and the diseases they cause. I would make flashcards of those as well. Be sure that you have them and put all that pertinent information we talked about, single-stranded or double-stranded, does it have spikes, that type of thing. All right, last slide for chapter 5. This is probably the shortest part of the lecture, but I do try to find places to stop them where it makes sense, all right? So some are longer than others. So the nature of viruses makes it pretty difficult to design an effective treatment. So we know that we don't have very good antiviral treatments and prevention. The best prevention that we have is vaccination, all right? Antibiotics, do they work on viruses? No, and this is a common problem. People go in, they're sick, they ask for an antibiotic, but if they have a virus, it is pointless unless you have a secondary infection. They do not work. Viruses do not work like bacteria. They use the host proteins and functions to copy themselves. So it is extremely difficult to design something that will affect them but not affect our host cell. So it makes them very, very hard to treat. Current antiviral drugs typically target steps of the life cycle. All right. Things like we have integrase inhibitors, which is going to be a class of HIV drugs that interrupts the ability of the HIV genetic info to be spliced into the host cell DNA. Okay. Much easier to develop a vaccine and prevent a virus than to treat a virus.